Hello, grade 12 learners of South Africa. Another session of life science brought to you by Mindset. And today we are going to be looking at nucleic acids. Now, we have to go back to the history of nucleic acids. I mean, you can't just jump in the middle of nowhere. We have to know where they actually came from and who found them, etc., etc. So here we go. Discovery of genes and DNA. First of all, we're going to look at our nucleic acids, and they were first identified in the 19th century. Now, these are things that they can ask you in the exam in multi-choices, for example. Then we have old James Watson Crick, at least James Watson and Francis Crick, and then Maurice Wilkin also had a part to play in the whole process. But you know, in a lot of biology labs and botany, zoology labs, science labs, you have the proper model of the DNA as it twirls around, or the double helix of the DNA. That model is actually called the Watson-Crick model because it was good old James Watson who came up with it, and Francis Crick. A lot of the theoretical work was done by Wilkins. So Watson-Crick model, remember that. So if they do ask you, it's an extra 2 marks for you in a multi-choice. Okay, and they won the award for DNA for their model in 1962. Long time, way before you were born. Then, in the late 1950s, Arthur Kronberg and Severo Okai, now he was a Russian, synthesized DNA, and later Okai was also able to decode DNA. Now that synthesizing of DNA, what he did was he worked with other Russian scientists, and you'll learn about what they did when we get to diversity, but they tried to simulate the Big Bang effect and create life from a huge big explosion, which is what one of the theories is, that where we came from and why we actually exist on this earth. Now, people, functions of the nucleus. Just use your common sense. You have done parts of this in grade 8, grade 9, grade 10, and grade 11. And when you did mitosis, you would, in grade 10, you would also have covered the functions of the nucleus. You would have done cell structure, again, functions of the nucleus. Here we go. Without this little nucleus, we cannot survive. Why? It controls cell metabolism. In other words, how quickly or slowly we make things or break things down in our bodies. You'll find people, skinny people, that eat loads of food. They eat like almost a ton of food a day. I'm sure you all know people like that. And most times it's boys, girls, that's something to be jealous about. Guys eat so much and they stay so skinny. But that means they have a fast metabolism. In order to have a fast metabolism, each little cell has to do its job. And remember, cells make up tissues. Lots of tissues make up organs. Lots of organs make up a system. And all the systems in our body make us the organism. So it controls cell metabolism. It is responsible for cell division. Mitosis and meiosis can only occur if there is a nucleus. If there is no nucleus, well, guess what? It cannot occur. Meiosis will occur in the sex cells, so in other words, the gonads, and your mitosis in every single somatic cell, which is a body cell in our bodies. Okay? It also controls the production of RNA. Now, without RNA, we cannot have protein synthesis. People, every single organism on this earth is unique. Not most unique, not very unique, because it can't be. Unique means absolutely individual. So when you speak English or when you express yourself, it can never be most unique. It is unique or not. So unique in itself means it is special. You sitting there, myself, even my children will have different proteins in their bodies. Your moms and dads will have different proteins to you. Your brothers and sisters will have different proteins to you. They may have very similar proteins, but no two organisms on this earth, except if they're identical twins, will have the same proteins. So, if there's no RNA, we can't produce proteins. And proteins control our bodies. Now, without protein synthesis, we can't have our proteins, and the nucleus determines the proteins that are going to be synthesized. Remember, synthesis means to make. So protein synthesis to make proteins. And then responsible for the transfer of hereditary characteristics or hereditary traits. Traits is a shorter word than characteristics, and it means the same thing. For example, this nose that I have, shame. My poor children all have a nose like this. So I passed this nose on to my children. It was a hereditary characteristic. I got this nose from my father, who got it from his father, who got it from his mother. Sure, can you imagine? Another woman with a nose like this. So everything that you are, your eye color, the shape of your eyes, the shape of your mouth, the way you smile, the shape of your toes, everything is a hereditary characteristic, and that information is stored in the nucleus of every cell. So it's almost like having a plan, and you know, like you have a plan for a house or a building. That plan and all the recipes are sitting in your nucleus. All that information is contained in the nucleus, and that is what makes you absolutely unique. You are special. So, responsible for the transfer of hereditary characteristics. Let's look again. Functions of the nucleus. It controls cell metabolism. It is responsible for cell division. It controls the production of RNA, because we need the RNA for protein synthesis. The nucleus controls protein synthesis, and it is responsible for the transfer of all hereditary characteristics. What exactly you are made up of. Okay, now, we're going to look at the structure of nucleic acids. Now, with the structure of nucleic acids, remember, your nucleus is made up of chromosomes. On the chromosome, you are going to have DNA, and the DNA contains the genes. So, we take the genes, they sit on a specific place on the DNA, on actually the nitrogenous bases or nitrogenous bases, so that you know how to spell. And that joins up to form the double strand, which is DNA. That wraps around the protein core of the chromatid, and the chromatids join by a centromere to form a chromosome. So, looking at nucleic acids. Nucleic acids make up DNA and RNA. Now, if I wanted to build a house, I would have bricks. And I would put one brick on top of another brick on top of another brick in rows, and I would build my walls. The same applies here. Your nucleic acids are the bricks that build your DNA. So, and your RNA. So here we go. You must know the structure of nucleic acids. Very, very important. The building blocks of a nucleic acid. The building blocks of every nucleic acid is a nucleotide. They will make up the building blocks, so lots of nucleotides will join to form one nucleic acid. Okay? Now, this is one nucleotide, and I can promise you now, any exam you have, they will in some formal manner ask you what a nucleotide is, or what a nucleotide is composed of, or what the building blocks of a nucleic acid is. It's going to be a nucleotide. People, Definitely 3 marks in any exam paper, and I promise you, if we look at all the 3 and 2 and 1 marks that you can get, and you add them all together, we will eventually have 150. And can you imagine your elation if you can get 150 out of 150 for an exam question? So remember this, we have nucleic acids, and nucleic acids are made up of nucleotides. They are the building blocks. So we start off with the pentose sugar. Now remember, a pentose sugar is a 5-carbon sugar. So this sugar will have 5 carbons, and because it has 5 carbons, it's called a pent 5. So pentose sugar, 5-carbon sugar. Then we look at secondly, the phosphate ion. Now this phosphate ion and the pentose sugar form what is called the backbone of the nucleic acid. So you would have, for example, a phosphate, pentose sugar, phosphate, pentose sugar, and we're going to do a nucleic strand or chain now, but they form the backbone of all nucleic acids. And then we have the nitrogenous base, and this nitrogenous base, well, it's pronounced nitrogenous, but you will be forgiven if you say nitrogenous, because nitro means it contains nitrogen because it's the protein part of the DNA. And the difference between carbohydrates and fats and proteins is that your protein molecule contains nitrogen, whereas your fats and your carbs don't. So nitro, because it's protein and it contains nitrogen, and genus, if you look at the word, let's do this. Genus, where do you think this comes from? It originates from the word gene. So it's telling you it is the nitro, contains nitrogen, it's a protein. Genus, it's what makes the genes, and your genes sit there. Okay. Lovely. Now this is a nucleotide, and what we have then is a whole bunch of nucleotides that are going to be put together, but they're going to bond at the nitrogenous base. That's where they're going to join. So we're going to have our phosphate and the pentose sugar, and a phosphate and a pentose sugar. They form the backbone of the nucleic acid. And where the joiners for DNA, for example, will be at the nitrogenous bases. Now, very, very important. Please listen to this. Take it in. Close your eyes. Picture what I've done on the screen here and absorb it. Because if you get this, you never ever have to learn it. And any question that you get where you have to apply your knowledge will be based on this, as far as DNA and RNA is concerned. So, focus. I need you to focus. Here we go. I'm going to go blue. We have our nitrogenous bases. That was part of the nucleotide that attaches to the pentose sugar. Let's just go back. Here, phosphate and the pentose sugar forms the backbone, and the nitrogenous base, that is what's either going to join if it's DNA or carry certain information if it is RNA. Now, nitrogenous base. Nitrogenous bases are made up of 2 groups. They're called complementary bases. Now people, when something is complements you, if you say these 2 friends are complementary, in other words, they look after each other, they complement each other. So we have 2 sets of complementary bases. Okay? We have the purines and No, man. And we have the pyrimidines. Okay? So, purines, pyrimidines make up nitrogenous bases. They are the complementary bases. Purines, pyrimidines. Now watch this. Pyrimidines, pyrimidines have a Y. And all the bases that fall under pyrimidines, you'll have cytosine has a Y and thymine has a Y. So this has a Y, this has a Y, and that has a Y. Okay? So nitrogenous bases, complementary bases. Purines, pyrimidines. Pyrimidines are made up of cytosine and thymine. Okay? Now, we're going to have thymine in DNA. But in RNA, there's only uracil. So uracil will replace thymine always if it is an RNA. So if you see any nucleic acid and it's only one strand and there are no T's, it will have U's in place. Why? Because in RNA, uracil replaces thymine. So let's recap again. Nitrogenous bases, we have grouping complementary bases. We have purines and we have pyrimidines. Pyrimidines have a Y in the word, and cytosine has a Y and thymine has a Y. But uracil is an RNA, and I mean a U and a Y are very similar. So that is how you're going to remember it, people. Then the opposite applies for purines. Purines, we have adenine and guanine. So A and G. Okay, now the way they're complementary bases. So your purines are always going to join with your pyrimidines, always. And in other words, your adenine and guanine is going to team up with your cytosine and thymine. And there's a very specific combination. Your purines, adenine, will always combine with thymine. So remember the word at. Okay? And your guanine will always combine with cytosine. So it's always C to G or G to C. And your adenine to thymine will always be A to T or T to A. But remember the word at. There's your combination. And what's left? The G and the C. Another way of remembering is that C is a little letter like that, and G is a C with a foot on it. So these two go together, and A and T go together. Your purines always have a Y, so thymine and cytosine, except for uracil, which we will find in RNA, and that starts with a U. All right? I hope you've got this. Nitrogenous bases, complementary pairs. Purines, close your eyes, pyrimidines. Your pyrimidines are divided into cytosine and thymine, which is in DNA, and uracil in RNA. Your purines are made up of guanine and cytosine. And how do they combine? Your complementary bases, your purines always combine with pyrimidines. So your purine, adenine, will always combine with thymine, and guanine will always combine with cytosine. If you remember this, you cannot get any questions wrong on DNA. Okay, now the structure of a nucleic acid. People, look at this. Here we've got the whole nuclear chain. Do you remember I said to you that we have the backbone? There's your phosphate, pentose sugar. Phosphate, pentose sugar. Phosphate, pentose sugar. They form the backbone of the nucleic acid. Now, remember the 4 different nitrogenous bases? We're going to have adenine, which is a purine, guanine, which is a purine, thymine, which is a pyrimidine, and cytosine, which is a pyrimidine. Why? Remember all our Y's. Okay, now if we draw a DNA, then we're going to have the same. We have phosphate, pentose sugar, phosphate, pentose sugar, phosphate, pentose sugar, phosphate, and etc. Now, attached to here, we're going to have a nitrogenous base. And it's going to be a complementary base. So, if that is adenine, then this side must be thymine, because it must be a pyrimidine. Guanine is a purine, so cytosine is the pyrimidine. Thymine is the pyrimidine. It will always bond with an adenine, which is a purine. Cytosine, which is a pyrimidine, will go with guanine, which is a purine. So it's always purine, pyrimidine, purine, pyrimidine, pyrimidine, purine, pyrimidine, purine. Pyrimidines always bond with purines. Adenine to thymine and cytosine to guanine. And I'm sure you're going to say, for heaven's sake, lady, move on. I'm stressing this, and you will see perfectly well why when I start going through the questions which I've got for you. And you'll be able to see, I needed to know that. And I'm sure by now you know it, which means you'll never forget it, which means you won't ever have to learn it again. Right, so this is a nuclear chain showing our 4 nitrogenous bases. Here is a strand of DNA. Now what they've done here is they've taken the strand, and if you look here, there's the guanine and the cytosine, adenine and thymine, thymine and adenine, thymine and adenine, guanine and cytosine. And what we've got is a weak hydrogen bond here in this region here. There. So between the thymine and the adenine, or the pyrimidine and the purine, there's a very, very weak hydrogen bond, and it can be broken very easily. That's why it's a weak hydrogen bond. So that when the DNA has to unzip for whatever reason, in DNA replication or protein synthesis, then that bond will break, and there are enzymes that will cause it to break. Okay, so here we have the guanine nucleotide. There's the phosphate, pentose sugar, and the nitrogenous base. Here we have the cytosine nucleotide, phosphate, pentose sugar, nitrogenous base. The adenine nucleotide and the thymine nucleotide. And we've got our weak hydrogen bonds there. Okay, people, so this would be a strand of DNA. Now look, what does it do? It's a double strand of nucleic acids. Okay, so it's a double strand. And it is twisted. We call the twisting, we say it is a helix. Helix means to twist. Okay, people, now we're looking at DNA replication. Now, you know, we've all got zippers. And if you take that zip and you pull it up, now if it's closed, and you pull the zip down, that is exactly how the DNA replicates, like a zip that opens up. So let's have a look at the theory, and then I'm going to show you the diagram. All right, with DNA replication, when you replicate something, it means that you duplicate it. All right, so it takes place to produce 2 new identical DNA, and that is the most important process here, because the DNA is first going to unspiral, because remember, your DNA is a double helix, so you've got this sort of setup. Okay, it unspirals so that we end up with the 2 strands of DNA clearly joined in the middle by the with the nucleotides. Now, the weak hydrogen bonds that hold the complementary bases together, well, they break open. Each strand of the separated DNA attracts a new, now look at this, complementary nucleotide. Now, when we look at those complementary nucleotides, remember that adenosine is always going to bond with thymine, all right? And
cytosine will always bond with guanine. Just remember at, and then the other two are the two that are left over. Now, the resulting DNA, that's the one that ends up, contains half of the original and then half, which is the new complementary part. It's an exact copy of the original DNA because you've got one strand that is the original, all right? And you are now going to have one strand that is the new. And then over here, we're going to have the original and we are going to have the new. So what have I got now? I've got an identical set of DNA. All right, this process is necessary for mitotic cell division. Now remember, with mitosis, you are going to end up with two identical cells. All right, always two identical daughter cells. So for that to happen, you need the DNA to be identical. So let's look at this process now. This is a diagram or schematic, at least a diagrammatic representation of the DNA. So here's my DNA, and what happens? It starts to unspiral, and as it unspirals, so your DNA starts to separate out. Now these little guys that come along here, this will be, maybe that's thymine, and that's another one there, and let's get another, okay, let's do this one. And let's give it a different color. And this little one here will be guanine, and that's another little guanine there. And what are they doing? They're matching up with their complementary bases. Let's make this one a different color, and I'm just looking if I can find any others. Okay, maybe cytosine there. And that's how they do it. So what have we got? We've got our DNA that has now unzipped. As it has unzipped, so complementary bases have come into this side, complementary bases have come into that side, and I end up with two identical DNA, DNA molecules. This one, the one on this side, will have the original left, and this one will have the original right. And then the left and the right of the new complementaries fit in. So what do you end up with? Here, you have your backbone, and there I have your new strand sits there, and your new strand sits there, and this is your old strand, and that's your old strand. And this is DNA replication.
Now, your RNA molecules, we have the following. We have the 3 types of RNA, and this is important. You've got the RNA that takes the message from the nucleus and the DNA to the ribosome. Now, rattle your heads and remember, when you did cell structure in grade 10, you would have learned about the nucleus and you would have learned about the ribosomes within the cell. These are all intracellular. So you'd have the ribosomes. And what was the function of a ribosome? The function of a ribosome was protein synthesis. That's what they did, protein synthesis. So a ribosome produced the proteins that were unique to you as an individual. You are absolutely special. You have very special DNA, which is a combination of DNA from your mom and from your dad. So one set of your DNA comes from your mother, and one set of your DNA will come from your father. Your 2 gametes, haploid gametes, they will join, and when they join during fertilization, they form a diploid zygote. Okay? Now, that diploid zygote has combinations of chromosomes that came from the father and the mother of that organism. Now, your DNA dictates who and what you are. The recipe for you as a human being, and every single characteristic, from the little wrinkles on your fingers to the shape of your chin, the shape of your nose, the shape of your mouth, all those characteristics are represented by genes that sit on the DNA at the nitrogenous bases. Your ribosomes need to know what specific proteins to make. Now, if you can imagine, I'm going to take this down. Here, you have your cell. And inside, I'm still using my magic pen. Apologies. Here you have your nucleus, okay? I mean your cell. Inside this cell, you are going to have your nucleus. Okay? Inside the nucleus, you are going to have the nucleolus. Okay, which is part of protein synthesis too. It also helps with that. And you're going to have a mush mash of looking like cooked spaghetti. That's going to be your chromatin network. And you'll know from mitosis in grade 10 that that chromatin network starts to lose water. And as it loses water, it starts to shrink. And when it shrinks, that's when you can see your chromosomes and identify your chromosomes. So your chromatin network looks like this. It's made up of, in human beings, 46 of these chromosomes. Okay, or 23 pairs of chromosomes. 22 pairs of chromosomes are called somatic chromosomes or body chromosomes, or we can also call them, when I say not body order, you can't call, I'm explaining them their body order, but they are called autosomes or somatic chromosomes. So they are for your body, 22. And one pair is your sex chromosomes or your gonads. So you'll have 23 pairs where 22 pairs are autosomes. I need to make this go down. Autosomes plus one pair, which are your sex chromosomes. And that in males will be XY, and in females will be XX. All right, and that's very important for when we get to sex determination. Now, this is our little cell. So we've got a chromatin network of the nucleus, the nucleolus. And then you had endoplasmatic reticulum, and you had smooth endoplasmatic reticulum, and you had other endoplasmatic reticulum that had ribosomes on it. And we have little ribosomes all over the show. And those little ribosomes are the little structures that are responsible for protein synthesis. So think of the ribosomes as, let's do this. They are like the kitchen. Okay? And the chromosomes that are sitting in the chromatin network, this is like the recipe book. And the recipe book to make antibodies, to make just about everything in your body. People, proteins in your body are used for every cell, because remember, cells have a cell membrane. And what is a cell membrane made of? Phospholipids and proteins. So every cell membrane of every cell in your body contains proteins, because the proteins are the protein channels, and that is to get the liquids and things from a lower to a high concentration against the concentration gradient in and out of the cell. All right, so all cells, as well as your chromosomes, are made up of protein. So cells are made up of protein. Every hormone in your body, except for your sex hormones, they're only made of fat. but every other hormone in your body is made of protein, okay? But your sex hormones, testosterone in males, estrogen and progesterone in females, that is made of fat only. But every other hormone is made of protein. So all cells are made up of protein or consist of protein. All hormones made of protein. Every enzyme in your body, every single enzyme in your body is made of proteins. So what do enzymes do? They lower the amount of energy needed for a reaction to take place. So we need enzymes. Everything in your body, all your metabolic functions are controlled by enzymes, which make them start and make them stop. Okay, so cells, hormones, except the sex hormones, enzymes, and every single antibody we have is made of protein. And the recipe to make all these specific proteins is found in the chromosomes. It's those chromosomes that say, hold on, there's that flu virus again. It was here a couple of weeks ago. Recognize it. The killer T cells get out, because those are your soldiers, and they get down there and they get out their bazookas and they start pounding away at that virus. But those bazookas are your antibodies. And those bazookas, to get those bazookas, the recipe for them sits here in your chromosomes, which sit in the nucleus of your cell. So that's when people say, we have to learn that your at least your nucleus, controls all cell functions and metabolism. That's why. It controls all cell division, meiosis and mitosis. That is why. Your nucleus is exceptionally important in your body. So we have our ribosomes, and we now are going to take the information that sits here on those chromosomes, okay? And we have to take our recipe, our information, to the ribosome so it knows how to do it. And what do? The ribosome is going to go from here to the little, at least the RNA is going to go from the chromosome, from the DNA, and it's going to take that information to the ribosome. And what is it doing? It's carrying a message. So we call it messenger RNA. So all of this to help you understand what we are looking at. So our three types of RNA. We have our messenger RNA. And because messenger is such a long word to write, when you have to use the word like twice or three times in a sentence, we say, no, we're going to call it mRNA. Now notice this is a little m and capital RNA, because RNA stands for ribose nucleic acid. So ribose nucleic acid. When that bond forms between the two chains of nucleotides, and it bonds to form DNA, it forms the hydrogen bond and loses an oxygen from the sugar. So it becomes deoxyribose sugar. So DNA is deoxyribose nucleic acid, and RNA is ribose nucleic acid. So RNA and messenger RNA, what does it do? It's formed by the DNA as a piece of 3 nitrogenous bases. Now people, very important, there are 3 nitrogenous bases. It's little groups of 3. And those little chains of groups of 3 are called a codon. Okay? The process to do this is called conscription. Now, transcription. Trans means to change, script means to write. So we are writing the recipe onto, or the recipe of the DNA is here in this codon. So think of a codon as a code. That's where the word comes from. And your transcription means to copy or write. And that literally is what the messenger RNA does. It goes to the DNA breaks apart, and it goes and it says, that nitrogenous base is a adenine and a guanine and a cytosine. So my little piece of 3 must fit in there. So if we look at our DNA, and the DNA strand opens, it unzips. Now remember, the nitrogenous bases are going to be sitting on here like that. Okay, let's just come up with letters. So we make this A, we make this T, we make that G, we make this C, we make that T. So on the opposite side here, A, the opposite side would be G. The opposite side for A would be T. Remember, A equals T and C equals G. And if you remember this, you can't ever go wrong. So, A to G, what am I doing here? Just wait. Where's my eraser? Okay, A to T. To T, and T to A. G to C, and C to G. Now, your messenger RNA is going to come in here, and it's going to say, okay, I'm going to work in pieces of 3. So it's going to come along here, and it's going to give the opposite. So instead of C, it's going to be a G. Instead of G, it'll be a C. And instead of T, it's going to be an A. So this here will now be my messenger RNA. And that code of 3 little nitrogenous bases, 1, 2, 3, the code is called a codon. And what has it done? It has copied as an opposite what we have here. So think of it as a negative of a photo. You know, in the old days, and your professional photographers will still do it. They don't use these little digital cameras like we all do. But in the old days, you went and you bought a film. And you put the film in the back of a camera. And then you took the photo. And when the film was full, you could take 24 or 36 photos. You'd take the film out, and you would take it in for them to print your photo. What the messenger RNA is doing is it's taking 3 nitrogenous, or groups of 3 nitrogenous bases. It's taking the code of the DNA, and it is copying them in a negative. So it's the opposite to. There's my messenger RNA. And this process, you must know, people, is called transcription. Trans means to move. Scription, it's taken the picture, the writing of it, and it is moving. Transcription. Right, now, we have the messenger RNA. The messenger RNA is done. We have the transfer RNA. Now we have to take this messenger RNA, remember, is going to now carry the message from the DNA, and it carries that message all the way here to the ribosomes. All right. The transfer RNA, written small t RNA, what this does is it goes throughout this cytoplasm in the cell. Oh man. It says all the cytoplasm in the cell here. Okay, in there, it's going to find a whole bunch of amino acids. Now, amino acids are your building blocks of proteins. Okay, amino acids are the building blocks of proteins, the same as nucleotides are the building blocks of nucleic acids. So, and the 2 types of nucleic acids, DNA and RNA. Nitrogenous bases that are found on every nucleotide, complementary bases are purines, pyrimidines. Purines are made up of guanine and adenine, and the pyrimidines are made up of cytosine and thymine in DNA, but uracil in RNA. Okay, so here we're going to transfer now this information that was taken here from the DNA, and it's going to take that information and transfer it from the cytoplasm to the ribosomes. Okay, that's the transfer RNA. So the messenger RNA copies it, the transfer RNA carries it to the ribosome, or at the ribosome, because their job is these amino acids here. And they've got to carry the amino acids to the ribosome. And then your ribosomal RNA, with a little r, what is their job? Their job is form the ribosomes and produce the proteins based on the information from the transfer RNA. Now, if we look here, and I mean, clearly we're going to explain this in far more detail when we get to the whole protein synthesis process. But if we just move this up, this codon from the messenger RNA is going to have ACG, all right? Your transfer RNA is going to go and find amino acids for the nitrogenous base, the opposite to that. So we're going to have ACG. So your transfer RNA is going to pick up amino acids that fit in with ACG, and your ribosomal RNA is going to now slot in here, and instead of A, it's going to have a U, because remember, in RNA there is no thymine, there is uracil. So the complementary base for adenine will be uracil, the complementary base for cytosine will be guanine, the complementary base for guanine will be cytosine. And if we go, so it's UGC, so if we go all the way back up here again, U, because remember, DNA has got T and RNA has a U, and GC. So it's identical. And what have you done? You've made an amino acid that is identical to the recipe that was on the DNA. So messenger RNA copies with a codon, which is a 3 nitrogenous bases, forms a codon, or we can call it a triplet. But I like codon because it codes what's there. So it's a codon in 3. The transfer RNA collects the amino acids in the cytoplasm, which have that combination, and your ribosomal RNA actually produce the proteins. So what do? They put the whole lot together so that the amino acids group together, and the amino acids, lots of amino acids have peptide bonds between them, and when you have lots of strings of amino acids, they group together to form proteins. Now, if we look at the differences between RNA and DNA, this is something they will ask you in an exam, okay? They will, without a doubt. In some formal manner, you are going to have to explain the difference between RNA and DNA. So we say, right, remember the tables? Very important. Write this table down and go and stick it up on the fridge or behind the toilet door or on the shower door so that you have to read it before you shower. RNA versus DNA. First thing we're going to look at is the sugar. So what have we got? Here we have a ribose sugar, and here we have a deoxyribose sugar. Deoxy, it's lost in oxygen. So ribose sugar, deoxyribose sugar. But in both cases, the sugars are pentose sugars, 5 carbon sugars. So the name of the sugar is based on the number of carbons. It doesn't lose a carbon, it loses an oxygen. So ribose sugar and a deoxyribose sugar. Pentose means it has 5 carbons. So ribose, deoxyribose. Next one. a single unwound strand of nucleotides. So we're going to have phosphate, pentose sugar, phosphate, pentose sugar, and we're going to have our nitrogenous bases coming off it. That's RNA. Okay, except in RNA, we have uracil, not thymine. Alrighty, so it's a single unwound strand. So here we're talking about the strand. Here, it has a double helix strand. Double means two. So we'll have two nitrogenous bases, two phosphates, and two pentose sugars that will join in each nucleotide. Okay, and it's a helix, means it is twisted. So single unwound strand, double helix strand of nucleotides. Then the strand is short. Okay, it's short. Why? We're talking about codons, and a codon is only 3 nitrogenous bases. And then when we look at the transfer, the ribosomal RNA, there we're going to have the anticodons, which will be the opposite to the codon. All right, so therefore your anticodons will be exactly the same as the piece of DNA that was replicated. So we have the codons, which are the opposite to the DNA code for 3 nitrogenous bases, and the anticodon, which will be the same as the DNA. So the strand is short. Here the strand is long. Here it only contains 3 bases in the codons and the anticodons, and here it may have many triplets. I use the word triplets there so that I would remember to tell you that a codon is also called a triplet. So it's triplet bases and codon bases, okay, or anticodon bases. So codon 3 pieces, and what is the biological term for 3 is tri. So a triplet, triplet. And RNA always contains uracil, and DNA in place of uracil will always contain thymine. All right? I want you to remember this. So, in DNA, we will have adenine will always bond to thymine. Thymine will only ever bond to adenine. Guanine will always bond to cytosine, and cytosine will always bond to guanine. Okay? Your pyrimidines, thymine, cytosine. Okay? Okay? And adenine, adenine, guanine, guanine, purines. All right, now, in RNA, you are going to have on your nitrogenous bases, on your codon, or your triplet, let's say you've got adenine, uracil, guanine. Your anticodon, which we're going to do in far more detail in our next session when we go through protein synthesis. In the anticodon, you're going to have the opposite here. So what is the opposite to adenine? It's going to be thymine, but remember we're talking about RNA, so there will never, ever, ever in RNA be thymine. It will be uracil. And instead of what will be the complementary base for uracil will be adenine, and the complementary base for guanine will be cytosine. So your anticodon will always be the same as the DNA. So what was on the DNA strand? It would have been T and A and C. But remember, this is RNA and this is RNA, and I'm writing it here so that you remember, because I know I did it there. So your anticodon will be the same as DNA except for the uracil versus the thymine. People, please, I really hope you've got it now. And if you've got it, and I've nagged like a real stuck record, that's great, because it will be stuck in your head and you'll never even have to learn it. Alrighty, now, question 1. Let's see how you go with these questions. Okay, with question 1 it says, the diagram represents a part of a molecule. Underline the operative word. What we're going to do now is I'm going to go through ways in which to, these are exam techniques. You never read a question without a pen in your hand. So, the diagram represents part of a molecule. Okay? Now we're going to have to check what molecule this is. So let's zap this up. Do we know what this is? Do we? It's a double strand. If it's a double strand with weak hydrogen bonds, it is going to be a DNA. There we go. Okay, study the diagram and answer the questions that follow. First thing you are going to do, and this is what you are going to write. You are allowed to write on those test papers or exam papers. They're yours. They get given to you so that you can read it and write down the answers for the questions to whatever's written there. Right now, you write on your paper, because what you're doing is you're saying to your brain, okay, brain, get ready. Access the file. Okay, so life science, pull out the life science file. Now, in the life science file, go to the folder called DNA or nucleic acids. Boom, now we've got nucleic acids. In that folder of nucleic acids, pull out DNA and nucleotides and chains and comparison between RNA and DNA. Pretty much everything we've done today. Now the file is open. Now you can access the information. Here we go. So, the diagram of a molecule, what molecule is it? You write here for yourself. It is DNA. Before you even look at the questions, the sub-questions, you write down the labels, always. I know that life science is slightly different from other subjects. Your diagrams are exceptionally important. First thing you do when you get a diagram is label it. Label, label, label first. That's number 1. So, what is number 1? Those are the weak hydrogen bonds. Okay? Number 2 is the round thing. What are those round things? It's the phosphate ion. Okay? Number 3 is the pentose sugar. It's a 5-carbon sugar, pent 5. Then, now we've got number 4 and number 5. How do we know what this is? But they give us clues. They say that this shape is guanine. So therefore, this shape here is going to be the same. So this will be guanine. Easy game. All right? We say here, this shape is adenine, which means this shape here will also be adenine. So what are they asking you? We know A to T and C to G. You must know this. Remember it. Stick it in your head. Put it on a post-it pad on your forehead. I don't care what you do. Remember, those are the only combinations. Okay? And where you have thymine in DNA, you will have uracil in RNA. So here we go. Adenine, what always bonds with adenine? Thymine. Now remember, we're looking at DNA. So number 4 is going to be thymine. And what always bonds to guanine is cytosine. And thymine and cytosine are pyrimidines, and adenine and guanine are purines. See, everything that I've gone over with you again and again, here we have it. So, here they tell you the key. A is adenine and G is guanine, okay? So we know that what's opposite is going to be cytosine, and what's opposite here will be thymine, all right? And this is part of a molecule, so now we go. Identify the molecule above, the molecule in the above diagram. We've already done that. Now, please, it doesn't matter if you've written it on your diagram already. You are now going to confirm that it's correct. So we are going to say it is DNA. All right, now, identify the molecule. It is DNA. And where do we find DNA? It sits on the thread-like chromosomes in the nucleus of every cell. So if they said identify the molecule and where is it located, always. But now don't give extra information. I'm just telling you so that you remember. Okay, it's DNA and it's found on the thread-like chromosomes in the nucleus of every cell. Okay, label the parts 1 to 5. We've done that. You see, now all you go back is you refer to your diagram and say, right, number 1, weak hydrogen bonds. Number 2 is a phosphate ion. Number 3 is a pentose sugar. Number 4 is thymine. Number 5 is cytosine. Thank you for coming. 5 marks. So far, oh no, they're only giving us, sorry, it's only 1 and 5. So 1 would have been the weak hydrogen bond and 5 was cytosine. Okay, 2 marks. Thanks for coming. What is the collective name for the parts number 2, 3, and 4? So if we look here, 2, 3, and 4. So what they're asking you for is a building block of DNA. And what is that building block called? I know you know. I know you know. It is a nucleotide. And what is a nucleotide? It is the building block of all DNA and RNA or nucleic acids. Because what is DNA and RNA? They are nucleic acids. Now, what is the significance of this molecule being able to replicate itself? What is the significance of that? Why do we want this molecule to be able to replicate itself? Think. What were the functions of the nucleus? Should we go back? Come here. What were the functions of the nucleus? And this will tell you what the benefits are. The benefits here are it controls cell metabolism. It is responsible for all cell division, both meiosis and mitosis. It controls the production of RNA in the cells. It controls protein synthesis, and it is responsible for the transfer of hereditary characteristics. There you go. Okay, now if that's what the nucleus does, and the DNA and the RNA is found in the nucleus, don't you think that that's what their functions are? Okay, so it's able to, now, we need to make all of this happen. How can we make that happen in a nucleus? Only if we are able to, and let's go back to our slide, our final slide that we were working on here. That was the function of the nucleus. So what is the significance of this molecule, which is DNA? All right, people, so let's look at this. We say, right, firstly, the DNA is going to produce or produces 2 exact or identical copies. Okay, so this is what's in it. They're exact or identical copies. That's why it's able to replicate itself. Then the second point, we're going to say that 2 chromatids are identical to that of the parent. And that is the significance of a molecule being able to replicate itself. This molecule of DNA. Remember, all our information, all the recipes for what we are and how we are and our personalities and our physical features, everything is coded, is written on that DNA. And if I want to produce a lip cell, which has got dry and flaked, and if I want to make a new one, I have to undergo mitosis. And that mitosis can only happen if the DNA and the DNA is able to replicate itself, for example, in mitosis. Alrighty, now our next question, question 2. And here you're going to see again, questions are repeated or similar. They are similar questions. Why? Well, the one question came from exemplar paper in 2008. The other one came from the February exam 2009. They are similar questions. And all the different exemplar papers and the different final exams and prelim exams and sub-papers in the beginning of the year, you're going to find there are questions that get asked every single time. People, you must know these facts, and that's why I keep on and on and on and going over them again and again. So here we go. Where is DNA found? Now think about it. It's found on the thread-like, because remember, that's what the chromosomes look like normally, is they thread-like. On the thread-like chromosomes, now if you didn't say thread-like, you wouldn't have lost a mark. But on the thread-like chromosomes in the nucleus of all living cells. And living is what's also very important. All right, what are the constituent groups of molecules that form a nucleotide? Now people, again, we answered that just now, we're going to answer it again. And the more you do it, the more you'll remember it, the less you have to worry about remembering it or learning it in the future. So, what are the constituent groups? We are going to have a phosphate ion, okay, a pentose sugar, all right, and the last one is a nitrogenous base. When a word is really long in life science, break it up into syllables. So you're going to say nitrogenous. And remember the nitrogen, because it makes proteins, or it is protein-based, and genus, it's the genes. So, and the fancy way of saying it is nitrogenous. But if you say nitrogenous, it's A1, because you will know how to spell it. And that's important too. So phosphate ion, pentose sugar, nitrogenous base, phosphate, our pentose sugar, and our nitrogenous base. Name the purines and the pyrimidines of DNA and RNA. The way I would do this is I would say, right, purines, this will be for DNA and for RNA. You would have guanine and cytosine. So what am I doing? Am I being silly here? Let's just do this again. You see, you need to focus. You need to focus. We would say in DNA and RNA, purines. And the purines would be guanine and adenine. Okay, now, let's use a different color here. In DNA, pyrimidines, we would have cytosine, come on, do you remember, and thymine. But in RNA, the pyrimidines are cytosine and uracil. Remember the uracil. Please remember the uracil. Okay? Almost finished. Now, let's see how on the ball you are. First thing you do, any question regarding nucleic acids and messenger RNA and any of those RNAs, what you are going to do is this. You're going to write A equals T and G equals C, and you're going to put a U here, and you're going to circle it so that you remember wherever RNA is involved, the U replaces the T. So here we go. Adenine is going to be uracil. Adenine, another uracil, because looking at the messenger RNA from the DNA. So it's going to be the opposite to the DNA. All right? And the uracil is going to replace the thymine. Cytosine will be guanine. Guanine will be cytosine. Cytosine, guanine. And a U, which is adenine. The A is not T. It's going to be U. And the thymine would be adenine. All right, people, have a look here. And I'm going to give you a minute. Do it yourselves quickly. All right? We're not going to show you the screen. I want you to just do it yourselves. So let's go. I'm going to do it at the same time, and then we're going to show you. So you ready? Okay, have a look. Have a look, have a look. Okay, it will be GGC. There's one triplet. TAA, which will be AUU, GCU, and UAA. You got it? All right, let's do the last one. Just write it down quickly. GCGAATTCA. All right, you ready? All right, let's go. I'm going to do it. You do it. Ready? Are you ready? There we go. Did you get it? Please tell me you got it. Great. People, we're done for today. I hope you've learned a lot. I know I've repeated something so many times, but you must know what a nucleotide is. You must know what purities are, what pyrimidines are, your complementary bases, your nitrogenous bases, etc. And if you just go and read through this, your X sheets are going to be out. Get hold of an X sheet, have a look at it, and you'll be fine. All right? And this, if you know what we've gone through here today, I promise you now, you will have nucleic acids and DNA and RNA taped. Next time, we're going to start with protein synthesis and how to make those proteins that are unique to us as human beings, to each one of us, and makes us so very special. We're also going to do DNA fingerprinting, and that is an amazing science. So it's almost worth going to study forensic science just for that, because I think it must be such fun. And CSI will have nothing on us. We'll have it taped. So have a wonderful time. Until I see you again, bye-bye.